Now these are the another set of important terms that you need to understand before going into the lesson. That is dominant. What do you mean by dominant trait? Allele that expresses the phenotype in heterozygous condition. For example, I told you you are giving capital T and capital T for tallness. And you are giving small t and small t for dwarf. Right? You might understand this better after we work out the monohybrid cross. So for now you understand, this is the capital T that we are using here denotes that it is a dominant character. Tallness is a dominant character in an organism, that is Pisum sativum. And dwarfness is definitely a recessive character. Allele that expresses only in homozygous condition. For example, if you have a capital T and a small t? Here also, the external feature of the organism you see here is tallness only. Capital T and there is a small t. Still it will be tall. Even in heterozygous. What do you mean by heterozygous condition? Because there is one capital and one small t together here, it is heterozygous. So, even in heterozygous condition, if tallness is expressed, then it is called as what? A dominant phenotype. And dwarf means it is a recessive phenotype. For it to express in an organism, that is dwarfness, it needs to be only in homozygous condition. Homozygous means what? Both the letters here you see should have small t only. So this is the difference between dominant and recessive. Let us go to the next one. Heterozygous means what? You have a DNA here and you have a capital T and a small t here. This is paternal chromosome, this is maternal chromosome. If you have this kind of arrangement, it is called as heterozygous. If you have on both the sides you have small t and small t or capital T, then you call it as what? Homozygous condition. If the alleles, that is the contrasting characters, are the same, then it is homozygous. If the contrasting characters are different, then it is called as heterozygous condition. Now coming to the next one, genotype means the genetic makeup of an organism. And phenotype means it is the observable physical characteristic feature of an organism. When I say pea plant is tall, that means I am talking about its phenotype. When I say the tallness is because of capital T or capital T small t, then what am I talking about here? It is the genotype of the organism. Coming to the next one, we will be using this term in the cross, that is F1. What do you mean by F1? It is the first filial generation, that is the first offspring of parent generation. And F2 means the offsprings of self-fertilization of F1 generation. For you to understand this, we need to go and work out the monohybrid and dihybrid cross. So let us see the first cross that was performed by Mendel, that is called as monohybrid cross. Now students, you should understand what is the meaning of monohybrid cross. Mono means what? Single. Hybrid means we are going to cross two different plants which are considered as parents. So you are going to select plants, that is plant A, and you are going to cross it with plant B. And these two plants differ in a single character only. That is why we call this cross as what? Monohybrid cross. There is a single character that is taken into consideration. So let us go according to your NCERT. So what is the first cross that he performed? Between two plants that differed in a single character, that is called monohybrid cross. And what is the single character we are talking about here is plant height. Now this is the single character. Now students, you all know what is the allele of plant height. What are the two different alleles of plant height? one is tall, and you are crossing it with the dwarf plant. Now what you have to do here, on the left hand side you should write these are the parents. So there are two parents, plant A, plant B. One plant is tall, another one is dwarf. Now we are crossing them. So what is the genotype of these two? If you write the genotype. Now for the parents I have given the phenotype. One is tall, another one is dwarf. The genotype will be, they are homozygous for that condition, so I am taking capital T, crossing it with small t, small t. The next one I should do is, the next step will be gametes. When you work out the gametes, there can be only one single possible gamete here. It is capital T. Here there is only one single gamete possible, that is small t. And what we are going to do is, we are going to cross them. When you cross, you get capital T and small t. This is called as the F1 generation. Now I already told you what is F1 generation. These are the offsprings that arise from the first generation when we cross the parents. Now what we are going to do is, we are going to do the process of selfing. What we are going to do, we are going to take the siblings here. That is, it is not one plant here, there might be many plants. So we are going to take two plants out of that F1 generation, and we are going to cross it now. When we cross it, what happens is that we need to work out the gametes again. There are two types of gametes for this. Capital T, small t. Always you have to write the gametes inside a circle. So these are the two possible gametes now. Now how do you cross these two to work out the F2 generation? For this you need to draw a Punnett square. A Punnett square looks like this. It was developed by scientist Reginald Punnett. So this is how it is. It is made up of 4 squares like this. So you have to bring this over here and this over here. Capital T, small t. Bring this over here, capital T, small t. So you have to consider one as male, either one, and here as female. So you write here, cross these two, you will get capital T, capital T. When you cross these two, you get capital T, small t. When you cross these two, you will get what? These two, you will get capital T, small t. And then when you cross these two, you will get small t, small t. Now students, listen. The entire monohybrid cross is done now. Okay. Now, what are the important things that you need to look for in a monohybrid cross is that whatever I have written on this side is very, very important, and also on this side. Your solution, your cross should be exactly elaborative like this. Don't worry about the question that is asked in the exam. There is a pattern to work out any cross, and this is the standard pattern for you to work on. If you do this, there will not be any mistakes at all. So now what we are going to do, the final one is that we are going to work out the phenotype and phenotypic ratio. Then we are going to work on the genotypic ratio. With this, the cross is over. So what do you mean by phenotypic ratio? How many plants are tall in this Punnett square? I want you to count this. Wherever you find capital T, so this is tall, this is tall, this is tall. So there are 3 tall plants. And how many are dwarf? There is only 1 dwarf. So this is what is called as phenotypic ratio. Coming to the next one, genotypic ratio. How many are homozygous tall? If you take homozygous tall, there is only one. So you write here 1 which is homozygous tall. How many are heterozygous tall? Capital T small t, capital T small t. So it is 2. So it is capital T small t. How many are homozygous dwarf? There is only 1. So 1:2:1 is the genotypic ratio for this F2 generation. So this is how you should work out the monohybrid cross. Now how do you work out the dihybrid cross? Here we are also going to take two plants only, plant A and plant B, but these two plants will differ not in a single character, but they differ in two characters. That is what is called as dihybrid cross. So let us take the parents now. So the parental generation will be this. One is round, yellow. What am I talking about here? The plant feature is the seed shape and seed color. So round and yellow are two dominant traits. We are crossing it with what? Wrinkled green. Now listen. What is the genotype we are talking about here? Capital R, capital R, capital Y, capital Y. You are crossing it with what? Small r, small r, small y, small y. So this is supposed to be the genotype. Now what should be the next step, students? It will be your gametes. How do you work out the gametes? There is only one possible gamete from these two. Here also one possible gamete from these two. So you have capital R, capital Y, then you have small r, small y. So these are the two possible gametes. Now when you join these two together, what is the resultant F1 offspring you get? You have capital R, small r, capital Y, small y. Remember, in monohybrid cross you had a capital T and small t. That is one character. Here we are talking about two characters, so you have capital R, small r, capital Y, small y. So this will be your F1 generation. Now what you are going to do is you are going to cross two of your F1 generation, two plants of your F1 generation to work out the F2 generation. So what will be the next step, students? You have to work out the gametes. So the gametes for these will be, please listen all of you carefully. You have to take these two first, and then this, and this, and this. So these are the possible gametes. So let us see capital R, capital Y. Okay, and then capital R, small y. These are the maximum possible combinations. We are working out the probability here. So you have small r, capital Y, and small r, small y. Same way, if you work out the gamete for this, you will have capital R, capital Y, capital R, small y, small r, capital Y, small r, small y. Now that you have the gametes ready, let us work out on the Punnett square and the F2 generation.